I hope you find today's talk interesting. Yeah, you might find it a little bit weird. That's all right. I think we in psychiatry need a little bit more weirdness because we've sort of been stuck in such a deep rut. And what I hope to do today is to show you not necessarily that these particular ideas are right, but that it's possible to have new ideas and actually try to rigorously test them. And we need to be doing more of that. You guys are ahead of us. Psychiatry, as I say, is not so good at that. All right, so if we think about, what are we really trying to do? I study depression, sometimes anxiety disorders, but at one level up, what we're really trying to do is to ask, how can we enhance emotional well-being? How can we have people have lives that they're happier with? Most days they wake up, they're glad to be alive, they've got a sense of purpose, they feel good, they enjoy their connections with other people, they contribute to the human family in ways that make other people thrive. How do we do this? And I want to suggest to you that there are two paradigms that are current. The big one, which I'm not going to talk about, and the small one, which I am going to talk about. They're not mutually exclusive, but they're different. And so most of what now has really gripped psychiatry is, I think, it's really a simple idea. So we know, that it's pretty rough being a human being, right? And that the world is far from perfect. People have known this since antiquity. The Buddhists think that you can only become an enlightened Buddha through the human realm because it has enough opportunities and enough intelligence that you can achieve Buddhahood, but so much misery that you actually have a spur to want to try to do that, right? And I think that really is the essence of it. So, if being a human being is inherently problematic, what are you going to do about it, right? And one answer, and again, this is the kind of answer that we like as human beings, is if being a human being isn't perfect, then what we need to do is re-engineer humans to be different than they are, right? That you're going to build something new. And the two ways that people are really focused on, and you can really see this in this huge NIH Human Brain Initiative, is basically genetics and robotics, right? Artificial intelligence and changing genetic codes. And all of these things are basically efforts to make human beings be different and sort of more perfect than we are. So that, if there was a SNP, which there's not, that increases your risk of depression, you take the SNP out so everybody be happy, If there is a brain circuit that is malfunctioning that makes you depressed, you're going to just change that brain circuit. Now, these ideas have certain sciences that they're most simpatico with: computation, bioengineering, genetics, robotics, artificial intelligence. Both this genetic idea, which has taken some bad blows in the last 10 years, and these ideas of sort of RoboCop futures, hang on the idea that human thought, human emotion, human behavior is essentially algorithmic, meaning that it's computable, and it's computable in some way that's tractable, so that we can essentially, all these ideas basically boil down to that. So this, of course, is where all the big money is. This is where the drugs are. This is where the machines are. The quest to perfect human nature has been at the core of most religions. It's a very, very old idea, very old drive. It's one way of looking at things. What we're interested in our research group, and I think many of my collaborators, is another way, which is basically, to say it simply, rather than trying to perfect humans by engineering them to be different, can we optimally understand what makes humans function the best, given what we are now? Can we optimize what we already are? Now, we can optimize what we already are while we're off discovering new ways to plant little chips and computers in our brain, but this way has a very different flavor to it, right? So that the sciences that it is associated with are different. It tends to, because human beings have always been evolved in these very complex environments, it tends to be more anthropological. It tends to be, of course, evolutionary, because we have to understand our evolutionary origins if we're going to really understand what type of creature, what type of animal we are. It requires the study of other animal species, and it tends to give rise to probabilistic, emergent, complex ideas, right? It's not saying that there's something mystical here. It's saying that this is more of an ecological approach where you get these very, very unpredictable and highly complex emergent phenomena that need to be understood. And so it gives a little bit of a different flavor, but this idea that what we really want to understand is even if humans have this huge ability for plasticity, what are core things that make human beings happy, successful, healthy? It's a very, very simple idea. So we are interested in trying to use these approaches to devise new treatments for depression. That is the essence of the talk, and that is the essence of our research group's interests. Now, part of the reason I think that we love computers and computer games and new fancy things and high technology is because although it has a dystopian side to it, especially people my age, there's this sort of sense that, you know, we're going to eventually perfect the world, that the world is going to transcend its imperfections, and, you know, everything is going to be beautiful. And the problem, though, is, and one of the reasons why I'm interested in these more ecological, cautious approaches, is that these sort of grandiose, engineered visions of perfection always come with a downside. So take a look at this. You all can see that. Would anybody want to spend their summer vacation there? Anybody want to build a vacation house there? Right? Anybody want to, you know, hike through that desolate hellhole? No, you certainly don't, right? So what is this? Well, this is a picture near where I grew up of what was once, of what was once the largest lake west of the Great Lakes, which was once home to the largest collection of Native Americans, but also large mammals in North America. It was the Serengeti of North America. Nobody knows this crazy lake in Central California, totally, you know, re-engineered to build LA, to build San Francisco. There's always this shadow side. If we forget who we are, where we came from, and we try to overly perfect things. So I have this core emotional conviction that we need to temper our grandiose visions of changing things with a vision where we also, before we do that, we really understand who we are and what we are, so we can better understand what's going to happen with the changes. Here's a painting of that same lake back in pre-Columbian times, just to make the point that another way of saying what I'm talking about is we have an interest in this possibility that we might be able to harness evolved human capacities, but also human needs, to enhance physical and emotional health, well-being now. We can, you know, if we can figure out what is the best of what we are and enhance that, recuperate it into the modern world, and if we can figure out ways to step aside somewhat from the worst of what we are, what is the worst of our evolved history, that this is a tractable way forward. Now, I'm not going to talk about this today, but one of the things that our research group does is study compassion meditation, specifically around this idea that a very core, hardwired thing in humans is this tendency to have in-groups and out-groups, and this powerful tendency towards tribalism, which you can see in Iraq today with Mosul and all these places falling. What I want to talk about today is not trying to mitigate negative inheritance from our evolved past, but how can we optimize and recover positive aspects of our evolved pasts, things that the modern world has separated us from, or that we no longer employ, that we might be able to bring back to our benefit to help us actually cope with these crazy, crazy conditions that we live in with modernity. So that is the sort of 10,000-foot view of this ethological, anthropological, evolutionary approach to trying to discover new treatments for depression. So one of the first things that this perspective, I think, challenges is this. Through research, we know that mental disorders are brain disorders. Now, this is the official statement of the National Institute of Mental Health. And on one level, makes sense, right? I mean, what other kind of conditions could mental illnesses be but brain disorders, right? We know that in some very real way, the brain is sort of the seat of consciousness. But I want to suggest to you that although this looks obvious, it's not. And in fact, it's dead wrong. And in fact, it's really wrong, and it's a really wrong, I think, direction for the field to take, because if you see mental disorders as brain disorders, you will only look in the brain for their treatments, right? And I think by doing that, not only are you missing the phenomenology of the disorders, because I think you have your conceptions of causality kind of mixed up, but you also foreclose prematurely on a bunch of really interesting and potentially powerful treatment options that don't specifically have to open up the brain like a medicine to get into them. And that's what I want to talk about for the next part of the talk. So, what do we mean if we say, well, if mental disorders are, if let's say depression, if it's not a brain disorder, well, what kind of disorder is it? So, if you are a reductionist, as I am, you believe that mental states, the mind, arises in some direct way from the functioning of the brain, right? That mind states can be mapped on in ways we don't understand yet onto brain states, and I deeply believe that. So in a way, we can say that the brain is the cause of all mental states. So the brain is therefore the cause of depression, right? No brain, no depression. As soon as your brain stops, no more depression, at least in this life, right? But that sort of proximal causality, I want to suggest to you, may not always be the most interesting causality, or the most important causality, or the first causality. And so what we have done in psychiatry and in mental health research is confuse. We haven't read our Aristotle. We've mixed up our types of causality. Just because the brain produces mental states doesn't mean that mental disorders are always result from the brain being damaged or the brain being wrong. I want to suggest to you that in very non-trivial ways, mental disorders can have clear causes outside the brain. So let me give you some examples of this by going up from the brain into higher levels of system organizations. So let's take the family as the first unit up from the individual brain. Then we'll talk about society, and then we'll talk a little bit about what you could call the biosphere or the ecosphere. So Myrna Weissman did a fascinating study a few years ago from a large data set called the STAR-D. It was a huge antidepressant treatment study. She rounded up a couple hundred women who were depressed, who had children under the age of 17. And everybody in this study went on an SSRI antidepressant, citalopram. They followed them for 12 weeks, and they looked to see who went into remission. So what Myrna Weissman did was she measured the depression of the mothers, but she also measured the depression and the other behavioral troubles of the kids, and looked to see whether does the mother getting better have any impact on the kids. And what this slide shows is that it has a massive impact. Mothers who took the medication and did not get well from their depression had kids who actually gradually had their symptoms get worse. Mothers who went into remission, taking the medication, had children whose depression had a huge improvement. And in fact, if you looked at the effect size of the mothers who went into remission, get this much improvement in their depression, their kids get this much improvement. So think about what that means, just in the simplest sense. Mother takes a biological object, the pill, puts it in her mouth, swallows it. Unless you think that the pill somehow magically floated out through her sweat or her breath into the eyes of her kids or something like that, the mother does the biological thing and the kids get the larger biological effect. Strongly suggesting, right, that in many cases, depression early in life can be caused in a non-trivial way by the depression of the mother. Right? That something outside the brain-body complex of the affected child is actually driving the child's depression in a causally powerful way. How do we know that? You turn off the cause. You correct the mother's depression, the kid's depression resolves. If the kid's depression came from a broken brain, how could fixing the mom fix the kid's depression? Think about that for a second, and that's going to come up over and over again. That's the level of the family. What about level of society? Well, this is a chart of happiness around the world. Happiness charts pretty nicely with material goodies, although it's complex. But a guy named George Brown, who we don't pay enough attention to in this country, has done these many really amazing studies in the 80s and 90s, looking at rates of depression in different cultures and rates of the types of stressors within those cultures that we know produce depression, right? So we know that certain stressors are much more depressogenic than others. I don't have time to go into what all those are, but what you can see there, you see the big one, that's Zimbabwe in the years that he did it. And then when you get over to the far ones, you're looking at certain European populations. And what's really cool is that, sad actually, as the types of stressors become more common in a society that promote depression, depression becomes more common in that society. So the same stressors that promote depression in individuals within a society are strongly associated with rates of depression across societies. So from this perspective, if you were this far out in space and you wanted to treat depression, well, you could try to give everybody Prozac, right? But that's only going to work in 40% of people. A more parsimonious way to treat depression would be to change the societal factors that produce depression. You'd get a massive drop in depression. Again, suggesting that although we think of these things as being sort of airy-fairy, they're not. They're powerful causal mechanisms that have clear biological antecedents and causal pathways. Las Vegas. No, it's not. That's not actually New York, New York. That's New York. Now, if we go to the highest level, the ecosphere, the biosphere, there are powerful data, and I'm not going to read all of these, to suggest that ecological factors potently impact the depression, cause people to do things when they're depressed. So, in the largest study done to date, they found that if you take air pollution, light conditions, temperature, sunspots, those four factors account for about 1/3 of all the rates of suicide in the world. So 1/3 of the variance can be explained by factors that reach all the way out to the sun. I had a big meeting one time, I said, how many people think sunspots cause suicide? People thought I was pulling their leg, right? Pollution, air pollution has been associated with depression and suicide over and over and over again. So is pollen. It's not just depression. There's very powerful data showing that the closer you live to a freeway when you're in utero or just at the time of birth, the stronger your risk of getting autism. And the effect size is about the same as having autistic people in your family, right? So we know that at an even higher level, these more diffuse factors they cause depression, and they open up possibilities for novel treatments. So dealing with air pollution would be a very effective treatment on a planetary level for depression. It's just not going to happen because it's not tractable. We can't do it. We don't have the will and the capacity to do it. But if you were God and you could really get a hold of things, this would be one of the first things you should do to treat depression. All right. Now what I want to do for the rest of the talk is talk about systems that we think of as being below the brain, starting with the body, and then talking about what I'm calling here the microbe sphere, and then it actually gets right back down into the biosphere itself. So, before we go into the body specifically, if factors outside the brain can be powerfully causal for depression, and I'm not saying anything that's exactly rocket science here, it's just that we don't take it seriously enough as a field, might those factors also be effective in treating it and maybe treating it better? Because we know that about 40–50% of people don't even get an adequate short-term response to antidepressants. And in fact, we all know this, and we know that antidepressants act as if the brain was just a bag of chemicals, right? Not the most topologically complex entity in the known universe. Of course the brain's not a bag of chemicals, and that probably explains why antidepressants to some degree have their limited efficacy. So we know that depression has to do with alterations of brain circuits. We know the alterations in the functioning of brain circuits. And so one of the rages in psychiatry now is to do deep brain stimulation. Right? So for $125,000, you can come in and have somebody put an electrode in your brain, and they don't put it willy-nilly. Most often they put it right below an area called the subgenual anterior cingulate cortex, which in many studies is hyperactive in people that are depressed. The idea of the needle is that a little bit of electricity essentially turns that area off. So it's a little bit like a punctate lobotomy, if you will. And of course, these deep brain stimulation studies are ongoing. They're very hard to do. They're very hard to assess. But some people seem to have a very, very powerful response. People that have had no response to antidepressants sometimes get a very powerful response, although there's some interesting data emerging that this deep brain stimulator may make people undepressed, but not make them happy. That's just really, really interesting. They may have accidentally fractionated out depression that way. So this idea that you can somehow, rather than just dumping chemicals on the brain, that you can topologically intervene with the brain to change the functioning of brain circuits is something I want to suggest to you that this larger anthropological, evolutionary perspective also supports, right? Because in fact, you know, the brain tends to get depressed when it perceives conditions in the environment that suggest that you as a human being are failing, right? That you're either sick and going to die, so medical illness all around the world, #1 risk factor for depression, or that you're a loser, right? That nobody likes you, that that your kids aren't going to succeed, that you got no status, that you've been shamed. These are the two royal roads into depression. How does the brain know that one is in that state? Well, how does it know anything? It knows it through the senses, right? We only know what the senses can reveal to us, and one of the fascinating things about sensory pathways is, of course, they're highly specific. Unless you have synesthesia, you don't taste sights, you don't smell colors. Each of these pathways is wired very specifically in the brain, and it's wired so that the content that it delivers is profoundly believable, right? People have a very, very hard time overcoming rationally what their senses are telling them. And not just the senses from the outside, but the internal senses, right? So as I'll show you, we've done a lot of work over the years showing that peripheral inflammation makes people depressed, right? That inflammation in the body is telling the brain. That's why when you got the flu, it's very, very hard to go out and go partying. It's because that internal sensory organ, which is the inflammatory arm of the innate immune system, it commandeers the brain, right? This raises an interesting possibility. Maybe we could use these sensory pathways as evolved deep brain stimulators. Rather than sticking a needle in somebody's head, maybe if we could figure out how to stimulate these pathways, we could convince the brain that it should feel good, that it's in a position where, that vis-a-vis the rest of the world, it should feel good about itself, right? So maybe we could just use these just like that. Now, I think any of these could probably work. What we've been interested in my career is the immune system, but what I want to talk about today is more recent work having to do with the potential that evolutionarily ancient thermoregulatory pathways that run from the body, especially the skin, we think, to the brain, have the type of specificity and the type of activity that they may be, to quote Chris Lowry, they may be nature's antidepressants, right? So what's the basis for thinking about this? Well, as I said in my little talk at the symposium this morning, I've made a career out of taking other people's ideas. I make no bones about it, you know. I've been fortunate enough to work with a group of just visionary, I think, genius people, especially people that work in the animal world. You know, I often say all I'm trying to do, as Jared said, is, you know, like put your animal models into the human key, right? Just so many animal models don't ever make it into the human realm, you know. So the idea that tweaking this thermosensitive warm temperature signaling pathway might have antidepressant effects originally actually started from Chris Lowry's work, not with temperature, but with a microorganism I'm going to talk about toward the end called Mycobacterium vaccae. But Chris has shown that if you take animals and heat them up, if you take mice and heat them up, that it induces an antidepressant effect on a forced swim test that looks like what you see when you dose them with an SSRI. And that really interestingly, the graph on the right, whether the animals got a little bit of an SSRI plus hyperthermia or placebo plus hyperthermia, the warmer their core body temperature was after that intervention, the more antidepressant-like behavior they showed. So there's this fascinating animal data to suggest that maybe hyperthermia has antidepressant properties. Well, you know, I said in the beginning that there's the RoboCop approach to sort of re-engineering who we are, and then there's this sort of anthropologic approach to trying to understand what are the things that, from an evolutionary point of view and from a human point of view, make human beings feel happy, undepressed? And one way of looking at that are what are things that we evolved to signal that we're in a good space. The other is, can we look across the history of humanity and identify things that people have done over and over again to make themselves feel good, that weren't bad, right? I mean, people, have drunk themselves to oblivion to feel good. We know there's some things that are really counterproductive. But it's interesting in the context of hyperthermia that so many of the world's indigenous societies discovered something like a sweat lodge or took up something like a sweat lodge from their neighbors. And, they never went to the sweat lodge to party. They always went to the sweat lodge for healing, for spiritual pursuits, to get ready for a battle, so for stress resiliency, right? I don't think it's an accident. I think that the reason they went to the sweat lodge was because, as I'll show you, they were activating pathways that run from the body to the brain that induce stress resilience, pro-social behavior, and have antidepressant effects. Now you can see this phenomenon in the modern world. Here are some, modern warriors in their sweat lodge. The sweat lodge isn't going to fix what's wrong with them, unfortunately. They need Moni's nutrition lecture, and hot yoga, right? Why the hell has it become such this crazy movement to go do yoga in unbelievably miserable heat? Oh, because it helps your body bend up in better positions. No, I think it's because of what I'm about to show you. So I'm not going to go into all the niceties of this. This diagram is very simplified, but what I want to propose to you is that there's a lot of animal data and human data to suggest that warming the skin, hyperthermia, activates a pathway that runs through the lateral parabrachial nucleus that has very specific effects on serotonergic nuclei. It stimulates two of them, the DRI and the DRLV, while missing a whole bunch of other ones that have effects such as inducing panic attacks that we don't want to stimulate. And that these two serotonergic nuclei between them, interestingly, project to areas in the forebrain in animals, the human equivalents of which are the most abnormal areas in depression, and also projects to the hypothalamus and to the raphe pallidus nucleus to essentially modulate thermoregulatory capacity. So let me say it simply. This is what I think hyperthermia does. There's evidence, a lot of evidence in fact, that people with depression have malfunctioning of this pathway. So this pathway exists in part so that when you heat up, mechanisms are turned on so you cool down, right? You have to keep your body and brain temperature constant if you want the computer to function optimally. And humans are better than any other animal at thermoregulation by far. There's a linear correlation across species between thermoregulatory capacity and IQ. So we think what's going on in depression is that this pathway, look at this like a thermostat, right? That the pathway is sort of, the thermostat set too hot. And what we think we're doing with the hyperthermia is essentially taking a hammer to the thermostat, banging the thermostat, sort of my type of homework, at home, and sensitizing the thermostat so that it becomes more effective. When it becomes more sensitized, more active, the two things you predict from this picture is there should be an antidepressant effect and there should be a lowering of body temperature, right? If this thermoregulatory capacity enhances, you're going to be more able to sweat, you're going to be more able to cool off, and your body temperature should drop. So if you stick people in a hyperthermia box, the two things you predict would be that their body temperature should drop and their depression scores should drop. Is that true? So we decided to try it out. And the first place we tried it out was actually this rather remarkable inpatient hospital in Switzerland, where people come for alternative treatments who are really in bad shape. So if you're depressed enough to come into a psychiatric unit but you don't want medications, you would come to this Aeskulap clinic in Switzerland. And we got 16 people with very high depression scores, 3 of which were on chronic SSRI treatment, 13 had no treatment, and we gave them all a single session of whole body hyperthermia. Put them in a chamber, heat them up to a core body temperature of 38.5, then leave them in the chamber for about another hour because the core body temperature continues to rise. So we sort of let them just sort of stew for a while, right? It takes about 2.5 hours, temperature in the box about 140 degrees Fahrenheit. As I told people this morning, I put myself in it so I'd know what I was doing here. I thought I was going to die. pouring sweat, huffing, puffing. You'd never stay this long in a sauna. So when you ask me would a sauna work, the answer is maybe if you stayed in it for 2.5 hours, right? This is extreme, it's very heat. It's mild whole body hyperthermia because you don't need anesthesia and you cap it at 38.5 core body temperature, but it's pretty hot. Well, what did we find? Sorry, the number should be 16. So in 16 people, remarkably, we measured their depression score right before they went into the hyperthermia box and 5 days later. And as a group, their depression scores dropped by almost 50%. Now, this is sort of what you see with an antidepressant at 8 weeks. This is this huge effect size, and it happened so rapidly. The 3 people that were on chronic SSRI treatment had no response whatsoever, consistent with some animal data that SSRIs, when given chronically, stop the brain effects on that circuit I just showed you of hyperthermia. So we saw this really striking drop in depression scores, especially in people not on antidepressants. And we saw a remarkably large drop in core body temperature. Exactly as we predicted, these two things really, really quite fascinating. Now, if people with malfunctioning or suboptimal functioning of the skin to brain to skin pathway, if that is suboptimally functional in some depressed people, you would predict that people with evidence of poor functioning in that pathway might benefit more than others if that pathway is improved, right? So, if you think X is causing depression and you've got a treatment that gets rid of X, people with the highest X are going to get the best response from the treatment, right? So one of the outputs of this pathway that would suggest suboptimal functioning is elevated body temperature. And depressed people as a group have elevated body temperature. But we predicted, and you would predict, that elevated body temperature, paradoxically, should be a strong predictor of response to hyperthermia. Why? Because remember what hyperthermia is doing. It's not really that it's making people hot. It's that it's stressing, stimulating, sensitizing the system so that they actually then cool off, right? It resets the dial on the system. The people that are the furthest off are probably the ones that are going to get the best benefit from the resetting. And in fact, that's exactly what we found. So, there was a nice correlation. The hotter people were, the warmer their core body temperature in the 24 hours before the treatment, the bigger antidepressant response they showed 5 days later. The other thing that was really fascinating was that the more people's core body temperature dropped from before treatment to 5 days after treatment, the more their depression scores dropped over the same period. This is what got the thing into the American Journal of Psychiatry, because, it's hard to imagine a placebo response. People can't control their core body temperature, unless they're like an Indian yogi or something. So, pretty interesting, right? But, of course, one of the challenges here is that there was no placebo. And maybe you'd get undepressed too if I stuck you in a fancy box and made a big deal about it. So we had to come up with a placebo treatment. And so we decided to really try to build this work out at the University of Arizona, where I am. Down in the left-hand corner, you can see our brand new Heckle device. This is a fancier model than what we used in Europe. It uses infrared heat, and $50,000, and one of these can be yours. And so what we devised for a sham condition was we put people in the box, and so the heat comes from these very bright orange hyperthermic infrared lights. So we basically developed fake lights that develop the same light but have no heat. There's a fan that turns on. We got a fake fan, and we deliver a little bit of heat to the bottom of the box through these coils. So people think they're getting warm. And interestingly, so I'm going to show you data, very preliminary data from the first 13 people. 7 got the active hyperthermia, 6 got the sham. 6 of the 7 people who got the active hyperthermia thought they got the active treatment. So one person got cooked half to death and thought it was fake. Yeah, who knows, man? Of the 6 people who got the fake sham treatment, 5 thought they got the active treatment. So it's pretty cool, right? It really works pretty well. The results are interesting. Here we can see active in red, sham in blue, and over the first week, looked at here over a number of days by self-report instrument, there's a huge antidepressant effect of the active treatment. And in fact, the difference between the antidepressant and the actual and the sham, the effect size is almost exactly the same as the effect size we saw in Europe. So early data suggesting that there's some kind of specific antidepressant response to hyperthermia that's apparent within 2 or 3 days. So it's different than antidepressants. There's not that lag. It's more like ketamine. There's this rapid antidepressant response. And then we've actually now got data out to 4 weeks, and you can see this is now with clinician-rated scales. At 1 week, with the Hamilton Depression Rating Scale, even with that small population, it was already statistically significant. And one thing we were kind of pleased to see was that I thought before I did this that what would happen would be that these guys with the active treatment would go down, but then they'd kind of, they'd come back up. We're not seeing that. Clearly, most of the active activity is here in the first week or two. That's where the lines have a different slope. But then there's this sort of non-specific improvement that continues, much like what we saw when we looked at this in the European sample. So we have this really interesting data, although the numbers are very small, that this may in fact represent a really viable, that if you heat the body, because the head is sticking out of the box, right? If you heat the body, it's activating something that produces antidepressant effects. Well, what could that be? One of the interesting things is that we've been measuring cytokine levels before and after treatment, and the treatment acutely looks like exercise. You get a huge increase in IL-6. You don't get any increase in TNF-alpha. That's sort of the pattern you see with exercise. And that increase in IL-6 correlates very strongly with both the amount of sympathetic activation that occurs as a result of the heating. So we think what's going on is the heating is acutely stimulating sympathetic nervous system activity, and that is then driving IL-6 production. And the more it does that, it's a marker of the effectiveness of the box, and you see this strong correlation with clinical response. And then, interestingly, over time, though, we now have some preliminary data that a week later, people's heart rate variability is actually improved with the box. So we think we're probably, like exercising, over time, probably inducing a more vagal control. All right, so that's part one of the box. Now, this is part two. You know, we consider that we think of ourselves as ending at the skin, and that's true. There's no doubt that things within our skin are much more, show more allegiance to us than things outside our skin. But it's not a hard barrier. And this is a fascinating study showing how some of these sensory pathways can be powerfully influenced, not just by our body, but by other people's bodies. What they did, they took one group of people, and they had half of them exercise, and they collected their sweat. And they had the other half jump out of an airplane for the first time, skydive, and they collected their sweat, okay? So you got stress sweat, and you got exercise sweat. Then they took a totally separate group of people and put them in a scanner and stuck the sweat up to their noses, had them smell it. They probably had to pay them extra for that. People could not tell which sweat was which. Nobody could differentiate it. But when people smelled the stress sweat, it activated the stress systems in their brain. Their amygdala fired up. Isn't that amazing? And when they smelled the stress sweat, people became more accurate at reading threatening facial expressions, right? So the sweat of other people that they'd never met, that they did not consciously know was stress sweat, operating now through another sensory pathway, olfaction, changes brain functioning. You feel freaked out, you don't know why, you know. It's because Chris has been stressed out and I'm smelling him, right? You know, and then there's this evolutionary thing where then it also, you know, if your buddy is stressed out, you better start looking around too, right? It makes sense, but it points to the fact that these sensory pathways, this is very powerful and diffuse. And maybe if these sensory pathways can change our social brain, maybe we can use that, and something like hypothermia might change social behavior. And I want to tell you that I don't think hypothermia is an antidepressant strategy. I think it's actually a pro-social strategy. So one of the cool things we've been doing is before and after treatment, we've been having people wear this little listening device for 2 days straight. And the device is silent, and silently, every 12 minutes, randomly, it turns on and records 30 seconds of whatever you're doing. So it provides this snippets of daily life that allow you to see how much are people alone, how do they use language, how do they talk to other people. It's called the Electronically Activated Recorder of the Ear by our colleague Matthias Mell. And he has shown really interestingly that the more people are with others and talk to others, the happier they are. But it's not just random talk. The more deep, meaningful conversations you have with other people, the happier you are. So we've been looking, what does one treatment with hyperthermia do, not to what people say they're doing, but to their objective, real-world behavior? And the answer is, they spend less time by themselves, they talk more to other people, not to themselves, they have less small self-talk, but they're much more affectionate towards other people, they show more gratitude towards other people, and they don't complain as much, and they get more done. Isn't that amazing? So it really does look like people were getting in those sweat lodges for a real reason. And it's not statistically significant yet, but as you might guess, there is a correlation between improvements in these measures and reductions in depression. So this really gets to the point that this business about there's a connection between psychological warmth and physical warmth that Matthew Hale was talking about. We really think it's true. If you really heat people up, you can change their behavior in ways that make them warmer to other people, and that almost certainly is going to be involved in their antidepressant responses. Now, okay, interestingly also, these things that we measure with the ear are very powerfully related to the immune system. So we've just shown in that big graph on the right that if you ask people, how stressed out are you? How many friends do you have? And then you look at their immune gene expression analyses using the system of this genius guy named Steve Cole. Self-report, no association. In those same people, the more deep conversations they have, the way they use their language, big anti-inflammatory association. So the fact that the hyperthermic chamber changes these real-world social behaviors may provide a really powerful endophenotype for understanding immunologic processes that are also linked to depression. Okay, so let me finish up by suggesting that there's even a deeper way of taking this non-brain-oriented approach to depression and applying it to how we think about treatments. So I've sort of suggested that depression is a distributed, diffuse phenomena that can arise from causes in the brain, outside the brain. But I think on a deeper level, at its core, depression is really a conversation that's gone wrong, or a conversation that's gone lost. Now, we know this in psychological terms, right? So many cases of depression occur because of our interactions, and I use conversation metaphorically here, our interactions with other humans, right? You know, you catch your wife in bed with some other guy, your boss tells you that you're the worst employee ever and demotes you. These are amongst the most powerful depressogenic things there are. I think equally important to our conversations with our conspecifics are our conversations with the microbial world. And I want to predict to you that in 10, 15 years' time, we're going to realize that how we deal with the bugs, and I don't mean spraying insecticide on them, I mean how we work out our interpersonal relationships with the microbial world is going to turn out to be one of the main sort of pathways forward in terms of dealing with chronic diseases in the modern world, including depression. And I want to suggest to you that depression in the modern world, to some significant degree, I think is a result of a conversation that's been lost with the microbial world. Why is this so relevant? Well, I'm not going to really talk about the microbiota this morning, but you folks probably know more than others or realize more than others that as you sit here this morning, only 1 out of every 10 cells in your body is a mammal cell, right? You know, you're mostly bacteria, right? You're 90% bacteria this morning. From a bacteria's point of view, you as a human being are sort of like a stalk. You know, when the bacteria got bored over billions of years and they wanted to kind of look around and think, they created us to kind of do their bidding. I'll show you some evidence for that. So we need to stop thinking of ourselves as individuals and need to start thinking of ourselves as collectives. And a large part of that collective are organisms with their own DNA, their own agendas, that are in a community with us. We are each communities, not individuals. And when you see it that way, it opens up some very interesting, it's good for a few beers late at night and philosophizing, but it's also got some really interesting treatment implications. The bacteria on the inside, the bacteria on the skin, right? So everywhere where the self meets the world, there's this bacterial component, is much connected with other bacteria that didn't inhabit the body, but across evolutionary time passed through the body in massive numbers. And this idea of the old friend microorganisms, it's a term cooked up by a colleague of me and Chris Lowry's called Graham Rook, one of the real visionaries of the hygiene hypothesis world. And the idea here is a very simple one. Most of what the immune system needs to do is not attack things. What it mostly needs to do is not attack things. Not the self, but also not food. And as it turns out, because we co-evolve with this massive burden of microorganisms that were not pathogenic, how do we tolerate them? Because if you attack everything, you're going to waste all your energy, you're going to destroy your tissues, you're going to die young, right? That's why inflammation kills us in the modern world. So over time, these organisms, saprophytic mycobacterium, certain constituents of the microbiota, parasites, certain chronic viruses, these old friend microorganisms over time, because they had to be tolerated, became the teachers of tolerance. Because evolution always wants to outsource things, right? It's like the American economy. If it can do it cheaper offshore, it will. And so over time, it looks like what's happened is that a key components of training the immune system in immunotolerance were shipped offshore to these microorganisms that are now gone, right? They've gone extinct from the modern world because of toilets and those crazy, hand washing and the carpets and medicines, especially antibiotics. We are profoundly separated from these sort of ancient travelers with us. And there's overwhelming circumstantial evidence that this separation explains a fair amount on a societal level of why there's been this epidemic explosion of all manner of autoimmunity and allergies in the modern world. Countries that go from third world status to first world status get this massive explosion within a generation of every autoimmune condition and every allergy. We think, we've argued in a number of papers, that separation from these old friend microorganisms may also help account for increased rates of depression in the modern world. And here is one mechanism and one line of evidence that this might be the case. So one of the core ideas around the fact that inflammation is associated with depression is the fact that when you put people in psychosocial stressors, their levels of peripheral inflammation go up. People with early life adversity have much higher rises than others. People that have risk factors for depression get this inflammatory response to stress. This is a study done by Tom McDade, started about 35 years ago. They recruited a whole bunch of mothers that were pregnant in the Philippines, and they've been following their kids ever since. And all through childhood, they took note, they recorded repeatedly how clean were the houses. Was there horse poop on the floor? Were there pigs in the kitchen? Were there chickens underneath the house? How often did the kids get diarrhea? Were they born in a rainy season? So they had this wonderful algorithm for sort of early life microbial exposure. Now what they've done 30 years on is they've drawn blood, looked at high sensitivity C-reactive protein, a marker of inflammation, and asked people, how stressed out are you? And what they found was that just like us, the people that were raised in clean environments in the Philippines, because some of the people, had normal houses, the more stressed out you are, the higher your inflammation. What the graph on the right shows is that if you lost a child, if you lost a parent during childhood, much higher inflammation if you had a clean childhood. But the red line says that if you had a childhood just rife with these old friend microorganisms, if you're stressed out now, there's no rise in inflammation. If you lost a parent, no rise in inflammation. This is really interesting data, and it's one of the core questions in our field is the association between stress and inflammation an ancient, evolved thing that all humans share to help them deal with wounding, or is the inflammation that we see with stress mostly an epiphenomenon of our separation from these ancient immunotolerogenic microorganisms? And upon that, much is going to hinge in terms of how we understand depression and the immune system. One of these ancient microorganisms that we've been very interested in studying, and Chris has studied it elegantly and repeatedly in animal models, but we have not yet done it in humans, is a saprophytic mycobacterium that was discovered by Graham Rook and John Stanford in Uganda. You give this to animals, and they get smart. Give it to them, put them in a maze, and they go running through the maze. Give it to humans that have stage 4 terminal lung cancer, and the people that have adenocarcinoma that get this plus chemotherapy live about 6 months longer. They collected behavioral data in this study and found that the people that got this Mycobacterium vaccae also felt less tired, less anxious, less unhappy. They felt cognitively sharper. It showed antidepressant properties. And remember I said I have no problem borrowing other people's ideas. Chris has graciously allowed me to share a couple of slides from his work with Stefan Reber. Fascinating, fascinating paradigm where they take mice, and some of them just sit in their cage over on the left, but the others enter this thing called this chronic stress paradigm, where essentially for a month, every day, they toss a big nasty intruder mouse into the cage with the little local guys, and the intruder beats the hell out of them every day, and they cower and they get terrorized, and at the end of a month they look like they have post-traumatic stress disorder. They got all these terrible anxiety symptoms. They get a lot of inflammation in their gut. So what Chris and Stefan did was, in the weeks before that long-term stressor, half the mice got repeated injections of a placebo, and half the mice got repeated injections of this bacterium, Mycobacterium vaccae. And it really looked like the Mycobacterium vaccae worked like a vaccine. Maybe not like a vaccine. It's not a long-lasting T-cell mediated thing. Maybe more like an allergy shot. But it had this long-term protective effect. So the mice that got the vaccae, the first thing is that they didn't act the same way as the mice that got the placebo. They stood up for themselves. And because they stood up for themselves more, they were more proactive in their coping, the big nasty mouse left them alone a little bit more. It's you can see some of the implications of this for something like maybe people getting ready to go off to war. The mice that got the vaccae also showed less anxiety-like behavior, and they didn't get the inflammation in their guts. It also blocked that through a mechanism that required T-regulatory activity. So this is really interesting animal data to suggest that these old friend microorganisms, who we've fallen out of conversation with, may in fact have effects on brain-body functioning directly relevant to mood and anxiety disorders. And so when I said that it's a conversation that's gone wrong, I really am not kidding. Here's evidence that I'm not kidding, all right? So in my last couple of minutes, I've been rhapsodizing about bacteria like they're really, or viruses, like microbes, like they're good things. But that's only half the story. And one of the things I think that's also going to become much more spotlighted over the next 10 years is the fact that these are beings with their own agendas. And when you have a conversation, it's not like talking to yourself. There's somebody else that's giving you some lip. They're giving you some pushback. Bugs do the same thing. And bugs have remarkable ways of manipulating their hosts. Rabies being the classic example, where many of these microbes in species can take over the brains and totally change the behavior of the organisms. Lest you think that this has nothing to do with us and things relevant to depression, beautiful study recently from Indiana. Took a group of people just like you, quasi-normal, had them wear a little ecologically momentary device that about 9-10 times a day for a couple of days would beep them. And every time it beeped them, they have to stop. You have to stop and say who you're with and what you're doing. So it provides a real-time assessment of your perceived social interactions, right? So they did that in a group of people. Then that entire group of people got a flu shot. Then they repeated that social assessment 3 days later and 14 days later. And now, a flu shot, 3 days after a flu shot, is when you'd be maximally contagious if you'd actually gotten the flu virus. And what they found was that people became amazingly more social 3 days after the flu shot than either before. Don't know why. Hey, you want to go out and party? I don't know why I'm coughing, right? We think that being social is a good thing, right? So does the flu virus. The flu virus is playing us for a chump, basically, right? We think that we're masters of our own ship, we're wandering around socializing, but of course, in some way that we don't understand, the implication of the study is that the flu virus is driving our behavior to help its own spread. And so this is another important factor in thinking about depression as a conversation. It's not just a brain disorder. It's not just located. When we go into this microbial world, we can't just look at them as sources of proteins. We have to think about them as entities that we're engaging in a conversation with. Game theory and things like this in the years to come is going to become much more relevant to our understanding of this. All right, so in conclusion, this is how we tend to think of things. We think that when it comes to depression, there's all sorts of external risk factors, and they cause the brain to change. The brain is the cause. I'm suggesting to you that while this is sometimes true, there's an alternative paradigm that I think is just as often true. So let's take one of these factors. Let's take inflammation. So we and others, this is from our data, have shown that if you take people here with hepatitis C and you measure their depression for 3 months, if they're just hanging around, there's no change. But if you study their depressive scores over 3 months of getting interferon treatment, which powerfully activates peripheral inflammation, you get a skyrocketing rise in depression. Right? So we know that peripheral inflammation produces depression. Therefore, it should be possible that if you block peripheral inflammation, or if you block inflammation, it should work as an antidepressant, right? So we actually did the study. Andy Miller and I recruited 60 people with medically healthy, treatment-resistant depression. Half of them got 3 infusions of a powerful blocker of the pro-inflammatory cytokine tumor necrosis factor alpha, TNF-alpha. The other half of the people got 3 infusions of salt water. And then we followed them for 12 weeks to specifically test the idea that if pro-inflammatory cytokines cause depression, blocking them should work as an antidepressant. And the second hypothesis was that the higher the levels of inflammation, the more of an effect you should see by blocking the inflammation with infliximab. So halfway through the study, me and Miller thought we were going to win a Nobel Prize. We really, one night we almost started weeping. We were so moved by how astoundingly awesome we were, We discovered a new treatment for depression because 50% of the people were having miracle cures. Miracle cures. These people had failed 4 or 5 antidepressants. They were going back to work. It was unbelievable. We broke the blind, and the infliximab and the placebo were exactly the same. 50% of the miracle cures were salt water. Something to remember, put your head in the ocean 3 times when you're feeling depressed. This is the power of placebo. But the point for today is, remember I said that people with higher inflammation should be the ones to show the response. We found that 100%. Here on the right are people that had high inflammation. On the left are the people that had low inflammation. High here is a CRP of 5. Look, low inflammation versus high, blue is infliximab. Huge difference. If you got high inflammation, infliximab is much more effective than if you have low inflammation. And if you have high inflammation, infliximab is much more effective than placebo. The reason these lines didn't separate, and what we didn't expect, is if you have low inflammation and you block it further, it's actually worse, much worse. Just like the antidepressant slide I showed earlier this morning. Do not give every depressed person anti-inflammatory. These data suggest that we know that at low levels, these pro-inflammatory molecules have effects in the brain that are salubrious, that are health-promoting. This is a really interesting finding. But for today's talk, the main finding is that infliximab is too big to get into the central nervous system. When people have done spinal taps, even in really powerful autoimmune conditions that disrupt the blood-brain barrier, it ain't there. So think about what this means. It means that if you are somebody who has high levels of peripheral inflammation and you block that inflammation in the body and you do not get into the brain, it works as an antidepressant. This is the first, you can treat depression with a drug that doesn't get into the brain if people have abnormalities of that pathway that the drug directly affects. The simplest explanation, I would argue, is chronic inflammation in the body is telling the brain, you're sick, you're gonna die, you're gonna die. Well, who wouldn't get depressed with that message all the time, right? You take the message away and all of a sudden the brain goes, I guess I'm not gonna die. Hey baby, you wanna go out and have some lunch? You start feeling like, right? So what this suggests though, is that rather than looking at these things as risk factors, one of the clear implications of the distributed, dispersed causal theory of depression is that in ways that are most relevant, the causes for the condition can reside outside the brain, and the brain and its different behavioral patterns, it may not be that there's something wrong with the brain. It may be that the brain is having a normal, evolved response to that peripheral signal. Lest you not believe me, the brain on the right is a summary of the most often found abnormalities in major depression. See that area with the circle? That's the dorsal anterior cingulate. Hyperactive shows up, and that's the big finding. Look at the one on the right. That's not a depressed person. That's somebody getting interferon alpha. Chronic peripheral inflammation stimulates the same brain area. You could not tell which was which. A brain scan cannot tell you whether there's something terribly wrong with the wiring down there, or whether that's a normal response to some sort of environmental signal or some sort of perception of an environmental signal. And this is really a powerful idea because it suggests, this explains why if you stop some of these more causal, further back causes for depression, the only reason people could ever possibly recover is the brain couldn't be totally broken, right? Or what difference would it make if you took away that cause? The real suggestion here is that I think in many cases, although the brain activity is, quote, abnormal, it may be an evolved normal response to something outside the brain that is the true causal factor for the disorder, and that those causal factors then become novel targets for the discovery of new treatments. This is a complicated way of saying this, right? Don't overestimate the brain. For more information, here we are. Thank you for your time. I hope you found this interesting, and thank you.